So this is the third video in the Mendal series. So far we have only talked about simple patterns of inheritance where it's just two traits for any character and the character is controlled by a single gene. This is not really the norm. Most traits are multifactored or have multiple alleles. And so we're going to try to get into some other kinds of Mendelian relationships and see how we would work with those. So one of the important things to remember here is that the rules of Mendel, the principles of segregation and independent assortment, are still true even in the complex patterns of inheritance. It's just we have to think about it just a little bit more. So what am I talking about here? I can give you some examples, like for instance, when one allele is not completely dominant over the other, where like the flower color, instead of either purple or white, and purple is completely dominant. What if breeding actually did cause blending? Another is when a gene has more than one allele, so 2, 3, 4 different alleles possible, and how would those interact? And then what if a single gene produces multiple phenotypes, like in the case of albinism, for instance? Some other possibilities are epistasis, where one gene completely covers up the existence of the other phenotype. One phenotype present completely covers up the other phenotype on a different gene. That's called epistasis. And what if being homozygous was embryonic lethal? Then you wouldn't get a 3-to-1 ratio, but instead the homozygous would be dead, and so you'd only get a 2-to-1 ratio. Let's try to discuss these a little bit. So complete dominance is when is what we've been talking about so far, where the homozygous dominant is got the same phenotype as the heterozygotes, and it is different than the homozygous recessive. That's called a complete dominance, and that's what we've been talking about so far with the purple and white flowers. But you could also have an incomplete dominance, where the phenotype of the hybrids is somewhere in between the phenotype of the homozygous recessive and the homozygous dominant. Like flower color, for instance. And then we could also have codominance, where two different genes are both dominant at the same time. And so the phenotypes would be separate for the heterozygous versus either of the homozygous. And a good example of that is blood type. Okay, so let's talk about incomplete dominance. So in incomplete dominance, we could have one character where the color of the flower is either red, that's one trait, or white, the other trait. And in homozygous dominant, it's red, and in the homozygous recessive, it's white. But in incomplete dominance, the heterozygotes don't carry either of the homozygous phenotypes. And so we can't really call one dominant over the other, but instead they're both expressed a little bit. And so that's why we've switched the nomenclature, instead of having a big P and a little p for dominant and recessive. Here instead we're calling it color, C for color, and it can be either red or white. And so in the monohybrid crop, or the first hybridization, the parentals have their true breeding phenotypes, but then in the first filial generation, we end up with all pink flowers. So they have both the red and the white, and that means that the heterozygote has its own phenotype somewhere in between. So this is called an incomplete dominance. It's expressing both red and white at the same time, and that blends to pink. So what would happen if we, because Mendel's rules are still true, what would happen if we did a mono-hybrid cross, where we crossed the pink versus the pink? So what would happen in the F2 generation? Well, what we see is still the 1 to 2 to 1 genotypic ratio, and again, the heterozygotes have their own phenotype. So the homozygous red would be red, and the homozygous white would be white, but the hets would remain pink. And so we would get a 1 to 2 to 1 ratio. So the phenotype more closely resembles the genotype. And here you wouldn't have to do a test cross to find out whether it was homozygous or heterozygous, because the heterozygote has its own phenotype. So that's incomplete dominance. And when you see this notation, you know that you're dealing with incomplete dominance. But in a test question, it'll usually say it's an incomplete dominance situation. So the dominant allele does not completely subdue the recessive allele. Alleles don't interact that way. But instead, the dominant allele is a protein that's being made, and the recessive allele is usually the absence of that protein. And so that's what causes the simple inheritance patterns. So for any character, the dominance or recessiveness relationship depends on at what level we're examining. Because if we were examining at the phenotypic level, then we have to say what color the flower is. But if we were examining on the genotypic level, because we can do that now with PCR and running gels, running DNA out on gels, we could say that the phenotype is the presence of the other allele. So it's important to understand that dominance and phenotype are two separate things. Okay, and frequency does not mean dominance. For example, extra fingers and toes is a dominant allele, but most people in America have 5 digits. So even though sixth finger or sixth toe is dominant, it's not the prevalent phenotype in the population. So it's important to understand that dominant does not mean most common. We can have multiple alleles with shared dominance or incomplete dominance, like in blood group. The presence of an A protein means that you will not have antibodies against the A protein. And the presence of a B protein means that you won't have antibodies against the B protein. So there are 3 possible alleles. There's A, there's B, and there's neither. So if you have, if you're homozygous A, or if you are heterozygote A nothing, you will have A protein, and so you will have no antibodies against A. However, you would have antibodies against B. So you would not be able to receive blood from a donor who had B blood type. Similarly, B could be either homozygous or heterozygous. If you are heterozygote AB, then you would have both A protein and B protein. You would not have antibodies against A or B, and so AB blood is a universal acceptor. They can take blood from A or O. On the other hand, if you had O blood, you couldn't take blood from anybody but O because you have antibodies against A, you have antibodies against B, and AB blood has both proteins. And so one of the nice things about O blood, though, is that it doesn't have any proteins on it. And because it doesn't have any of these glycoproteins, it will not activate the antibody, the A antibodies that B blood has, or the B antibodies that A blood has. And so O is often called the universal donor. So it's important to understand that this is a codominant situation, codominance with multiple alleles. And so there are 6 different genotypes that you could have and 4 different phenotypes that you could have. Pleiotropy is when one single gene has multiple effects. It causes phenotypes in a bunch of different places. For instance, cystic fibrosis is a chloride channel that I talked about back in cell biology, in the cell chapter 6. And that chloride channel not only affects the lungs, which is what I talked about, but it also affects the person's ability to digest food because there's also mucus in those bile ducts and also the person's sweat glands. So pleiotropy is when one gene has multiple effects. There could also be another interaction between 2 or more genes, and that's epistasis, stand upon. Stasis is stand, epi is upon, so stand upon. So an example of that that's always given is in Labrador retrievers, you have 2 different independent genes. One is for coat color, and it comes in either the dominant black or the recessive brown. And then there's a second gene that determines whether pigment will even be deposited in the hair. And so when we do a dihybrid cross on this, we have the heterozygous versus the heterozygous, so 2 black babies, or black parents, and when they are crossed, instead of having a 9:3 to 3:1 ratio, we have a 9:3 to 4 ratio, because the dominance of black is seen here in the 9, and the recessiveness of the brown is seen here in the 3, but when you are homozygous for no coat color, it doesn't matter if you're not putting in brown or you're not putting in black, you're not putting in any color. And so those are where the blondes come from. So these are homozygous and heterozygous for black, but they get no coat color. And this is homozygous, or it's homozygous recessive, so it should be brown, but it has no coat color also. So instead of a 9:3 to 3:1 ratio, we get a 9:3 to 4 ratio. Polygenic just means that there's a lot of variability. This is common in human skin color. We also need to understand that in addition to multifactorial, the environment also has an effect.